So today we're still in the adaptive immune system. Remember, the adaptive immune system is very specific, and it includes the cell-mediated immunity and humoral immunity. We've already started talking about cell-mediated immunity, and that's where the T cells are coming into play. With humoral immunity, we're going to be talking about our B cells. So humoral immunity revolves around the B cells, and it refers to something floating around in the blood fluid of the body that's too small to see, and that's going to include antibodies. The cellular and humoral immunity are working hand in hand. They're not independent. The term humoral immunity is describing the pathway by which antibodies are produced by our B cells to target exogenous antigens. What are exogenous antigens? Those are going to be coming from something outside of ourselves, like bacteria. When macrophages engulf exogenous pathogens, they're going to digest them within lysosomes to release their antigenic fragments. And those fragments are presented on special surface receptors. Remember, those are the MHC class 2 for something foreign. The antigens are presented next to helper T cells, and those are going to secrete cytokines to activate the appropriate B cells. The specific B cells then are going to divide and differentiate. This is called clonal selection, where they just create identical copies of themselves. And they're going to then form antibody-producing plasma cells. So the key components are B cells, antibodies, which we'll get into next, the antigen-antibody interaction. The binding of an antibody to an antigen is highly specific, like a lock and key. And that binding can either neutralize the pathogen directly or take it for destruction by other immune cells. It also includes the complement system. That's a group of proteins that work in concert with antibodies to clear pathogens from an organism. And those proteins can directly cut up pathogens or facilitate their phagocytosis by immune cells. It also plays a role in inflammation, and it helps bridge the innate and adaptive immunity together. So what's the process of humoral immunity? It's all going to start out with antigen recognition. It starts when a B cell encounters its specific antigen. This antigen is typically a foreign protein or polysaccharide on the surface of a pathogen. The B cell receptor, which is basically a membrane-bound antibody, binds to the antigen. After that binding, now the B cell is going to be activated. That usually requires additional signals from helper T cells, which release cytokines that further stimulate the B cell. Then we get clonal expansion and differentiation, where the activated B cells undergo clonal expansion. That's where they're going to rapidly divide to produce a large population of identical cells. And then those cells are going to divide again into two main types, the plasma cells and the memory B cells. Our plasma cells don't live very long, and they secrete tons of antibodies, but the memory B cells persist in our body, and they're going to provide us with long-term immunity by responding more rapidly and effectively on re-exposure to that same antigen. Finally, antibody production and pathogen elimination. The antibodies produced by plasma cells are going to circulate throughout our bodies, binding to their specific antigens. And this binding can neutralize them directly or mark them for destruction by other immune cells. The complement system may also be activated. enhancing the pathogen's elimination. So humoral immunity is very important for protecting us from extracellular pathogens. Clinical implications: vaccines are designed to stimulate humoral immunity by introducing an antigen in a controlled manner, prompting the body to produce antibodies and memory B cells without causing disease. In some cases, the humoral immune system can mistakenly target our own tissues, leading to autoimmune diseases. There can also be a problem with deficiencies in humoral immunity, and that can make us more susceptible to infections. Now, viral infections are very important. Without certain basic childhood viral infections and exposures like chickenpox and measles, the immunological self-tolerance does not develop. Our immune system needs to be challenged. Otherwise, the immune system blows back on itself. Adaptive humoral immunity, T helper cell 2, is set with challenges. And if they're not present, then that part of the immune system blows back on itself. Now let's look at B cells, B lymphocytes. This image of a B cell gives you an idea of how many different types of receptors there are on the surface of a B cell. B cells mature in our bone marrow, which can then produce antibodies, and they can make up to 1 trillion different antibodies to 1 trillion antigens. However, one B cell can only detect one antigen. When they detect an antigen, they differentiate into plasma cells in blood, lymph organs, and inflammatory sites, which throw out antibodies, which either isolate or kill the bacteria. And then, as we mentioned, they can differentiate into long-lived memory cells. The memory cell's not there to fight. Instead, it's going to run off and hide until the infection's over. But the next time it detects that same infective agent, it's ready. B cells produce antibodies, or immunoglobulins. They're called antibodies when they're circulating for antigens. Antibodies and immunoglobulins are terms that are used interchangeably, but technically they're antibodies when free-floating through the blood, and immunoglobulins when they're actually stuck to the surface of a cell. Also, when they're free-floating in the blood, we call them serum glycoproteins. A vitamin B1 deficiency impairs the maintenance of B cells. Now, in this section, I'm going to invite you to look this up in the database to go through all these very interesting details. By the way, today when I used the search box to look up humoral immunity, it came right up. So sometimes the search box works great. Go ahead and use it. If it's not working, use the menus. The activation of B cells usually occurs in our lymph nodes, where there are a large number of B cells. Dendritic cells flow through lymph channels, carrying antigens to present to naive T cells. And here's an illustration. This comes from that video I asked you guys to watch from Dr. Bean several weeks ago. Here we see a dendritic cell, and that little arm there is the MH2, and it's presenting an antigen to a naive T cell, that's T cell 0. It's got an arm that it can connect with that antigen to learn about it. And then there are all kinds of other components, other kinds of cytokines circulating here that are going to influence what happens to that naive T cell after it connects with the antigen. If interleukin 4 happens to be present, then it's going to turn into a Th2 helper cell. And that cell, again, with the presence of interleukin 4 and a few other components from the complement system, it can then activate this B cell, and you see there the MH2 arm again. All these things are connecting up. Now the B cell is finally ready to connect to the antigen here on this pathogen to either destroy it or tag it. This section gives detailed breakdown of what's happening there. An activated B cell becomes a plasma B cell, which starts producing antibodies. So we have some studies here that you can look at. Now, we did speak briefly about herbal B cell inhibitors. When would you consider using those? Herbal B cell inhibitors are being explored for their potential to modulate the immune system naturally. They can offer a complementary or alternative approach to conventional treatments, especially for individuals seeking natural remedies or those who experience adverse effects from synthetic drugs. Now, when would you want to use a B cell inhibitor? For cancer treatment, B cell inhibitor drugs are primarily used in the treatment of certain types of blood cancers, like chronic lymphocytic leukemia, small lymphocytic lymphoma, acute myeloid leukemia. Also for certain autoimmune diseases. B cell depletion therapies, which include B cell inhibitors, are used to treat autoimmune conditions like lupus and multiple sclerosis. So herbal B cell inhibitors have shown potential in treating certain types of lymphomas and overcoming resistance to conventional therapies. And then in this section you can see an explanation of their use. Come into the database to read all of this. And here we see one of the primary compounds would be gambogic acid from the Chinese herb gambogis. You can click on that to learn about that herb. Come in here and learn about considerations for use. Common herbs with B cell inhibitor properties: turmeric, green tea, ginger, ashwagandha, licorice. Here we can get into the mechanism of action and considerations in safety. Before starting them, it's important to consult with a healthcare provider. You want to make sure you have good quality in sourcing of the herbs. Some herbal remedies interact with conventional medications. We already know that, so you check the cautions. And then individual variability is always a factor. All right, so next, let's move on to antibodies, immunoglobulins. Of course, I suggest you watch any of these videos to get started with this, but as we mentioned, immunoglobulins are also called antibodies. Once an antibody is attached to a cell, it's called an immunoglobulin. They're glycoproteins, and they are produced in response to bacteria, viruses, other microorganisms, and any substance that the body recognizes as harmful. They have a Y shape, and what they do is tag the bad guys. We've already talked about how B cells produce them. The blue triangles represent epitopes found on an antigen. Those could be a polysaccharide or a peptide, and they help the antibodies to tag them. So we see the Y-shaped antibody. There's the antigen. It's going to have these little epitopes here, and after being tagged by the antibody, the antigen generally comes under attack by other cells of the immune system, like killer T cells. And for the different types of antibodies in the database, you can click on immunoglobulins. So most human antibodies are polyspecific. They hit multiple targets. Antibodies are far more complex and numerous than we ever dreamed was possible. Human antibodies are mostly polyspecific. Autoantibodies is a misnomer indicating their potential for collateral damage. The most likely trigger for persistent antibody production in humans are the microbes composing our human microbiome, and the failure of innate immunity will lead to energetic stimulation of and hence autoantibody production. Stimulation of the innate immune system against the microbiome appears to control human antibody autoantibody titers. Now, this is a relatively new study. Bad antibodies made good, the immune system's secret weapon uncovered. Research revealed how a population of bad antibodies in the immune system, and they're usually silenced because they can harm the body, those can provide crucial protection against invading microbes. Now, this was a study carried out on mice. The bad antibodies are known to react against our own tissues and cause autoimmune disease. And for this reason, it was once thought that they were discarded by the immune system or that they were made inactive in the long term. However, new findings show for the first time that bad antibodies go through a rapid redemption process and are activated when the body is faced with a disease threat that other antibodies can't tackle. As a result, the redeemed antibodies no longer threaten the body, but instead become powerful weapons to fight disease, particularly diseases that evade the immune system by disguising themselves to look like normal body tissue. We're not going to get into autoantibodies right now, but if you look over on the left-hand side of the page here, you'll see we have a whole section on autoantibodies. We'll probably take those up much later. But for now, let's click on immunoglobulins. We've already talked about IgA, and today we're going to talk about IgE, immunoglobulin E. It's found in trace amounts in our serum. It's the least abundant immunoglobulin in serum with a half-life of 2 days. It's crucial because it plays an important role in allergic reactions, and it's also important in defense against parasites, especially helminths. That's a type of worm. So IgE are antibodies produced that are related to histamine and allergies. It's also aimed, as I said, at attacking large parasites. Mast cells are covered with IgE antibodies. Waiting. What are mast cells? Mast cell belongs to our innate immune system. It's also called a granulocyte. It's one of the granulocytes. It contains lots of little tiny granules of toxic chemicals that when released will kill pathogens. They also can harm body tissue. And here we can see a mast cell, and that's IgE, immunoglobulin. Remember, immunoglobulins, when they are attached to a cell, that's IgE immunoglobulin just sitting there waiting. Our mast cells play an important role in our immune system, but they're also troublemakers. They're extremely sensitive, and they're responsible for allergic reactions and even anaphylaxis. They get started in the bone marrow as a progenitor. They don't mature there. The progenitor is going to travel to our peripheral tissues. In fact, mast cells are restricted to tissues. They don't circulate in our blood. They're present in all of our connective tissues, but concentrated around blood vessels, the nerves, under all the external lining of the body, and under all the internal lining of the body. Under all the mucosal lining, there are a lot of mast cells. In terms of initial sequence of activation, mast cells are among the first to recognize danger signals relayed from our external environment, and that's because of their strategic location at the major body surfaces, including skin, the lining of the lung, and gastrointestinal tract. Once activated, they help orchestrate the early phases of our innate immune response, promoting recruitment of eosinophils and basophils, we'll cover those later, that follow inflammatory and chemotactic stimuli originating from inflammation. Mast cells, eosinophils, and basophils are activated more or less simultaneously in many diseases, including infections, allergic and autoimmune disorders, and cancer. Here we can see a mast cell with its IgE attached to a receptor. And then if you look over towards the right, an antigen connects with IgE. And once that happens, degranulation occurs. That means all those little chemical-laden granules lodged inside the mast cell explode, releasing a lot of chemicals. They're recruited during acute inflammation because the histamine is one of the chemicals in those granules that's released. Histamine causes vasodilation and increased vascular permeability. If the acute inflammation doesn't take care of the problem, these mast cells just continue releasing these molecules, resulting in a chronic inflammation process. They release not only histamine, but also heparin, and they are always releasing heparin in tiny doses. Heparin from mast cells appears in the blood to prevent clotting. Even if there's some microscopic clots being formed, these tiny doses of heparin helps prevent coagulation. Okay, so this is just a quick intro to mast cells. We're not going to spend a lot of time on them today, but we will come back to them as we dig into allergies, especially food allergies, and also certain mast cell disorders. But for now, we're just going to jump back to immunoglobulin E. You've seen how it plays an important role in what's going on with the mast cells. So how does it work to get rid of parasites, specifically worms? When there's an invasion of a parasitic worm, the T helper cell is going to secrete cytokines like interleukin-4 and stimulate B cells to secrete immunoglobulin E antibodies. Those antibodies rise in our serum, and then they coat the surface of the worm by binding to their surface antigens. These bound immunoglobulin E antibodies are recognized by another one of our innate immune cells, the eosinophil. This recognition is mediated by a specific receptor. I'm just going to call that, I don't know if this is the right pronunciation, FCER. I'm just going to call it FCER, receptor present on the eosinophil. These receptors recognize and bind the FCER region of immunoglobulin E antibodies. Once bound, they undergo degranulation and release the granulocyte content that destroys the worm. And here you can see a diagram of that. There's your eosinophil, here's your immunoglobulin E. It connects to that. We zip over here, there's the worm, they all connect together. The molecular contents of those granules is released and destroys the worm. Okay, allergies. So we know that sensitive or allergic people recognize certain harmless substances, allergens, as dangerous when there is an initial or first exposure to an allergen by B cells. Once activated, these B cells differentiate into our immunoglobulin E producing plasma cells. The immunoglobulin E antibodies attach themselves to mast cells and also basophils by their FCER regions. Now, the antigen binding sites of these antibodies are free. When there's a second exposure to the allergen binds to these antigen binding sites of immunoglobulin antibodies. This binding of allergen to immunoglobulin antibodies results in a cross-linking of immunoglobulin E antibodies. And it's that cross-linking that sends a signal to the cell and results in degranulation. So it's through the second exposure. A result of the degranulation is there's a rapid release of preformed mediators from the granules, for example, histamine, heparin, etc. And these mediators further result in vasodilation, increased vascular permeability, smooth muscle contractions, etc. So if you have an allergy, your immune system overreacts to an allergen by producing immunoglobulin E. Those antibodies are going to travel to cells that release chemicals causing an allergic reaction. And that usually causes symptoms in the nose, lungs, throat, or skin, but also the gut. I'm going to invite you to come in the database and read up on this section about the role of the immune system in IgE regulation. Just a few quick points here. The balance between T helper cell 1 and T helper cell 2 cells is crucial for determining allergic sensitization. So when we're struggling with allergies, obviously one area that we might want to look at is how much immunoglobulin E is floating around to trigger our mast cells. And is there a way to inhibit production of so much immunoglobulin E that's natural? You know, a lot of people take antihistamines to counteract symptoms of allergies, but remember, histamine's only one of the molecules that's released from mast cells that can cause problems. Certainly limiting it is helpful, but it turns out it's not the full answer. There are other routes that we can take to deal with allergies, and one of them is inhibiting IgE. So let's take a look at that and see what can be done. So as we've seen, immunoglobulin E plays a central role in allergic diseases, and understanding the network of interactions between it and its receptors opens new opportunities for therapeutic intervention. The interaction of IgE with its high affinity receptor, FSIR1, is directly implicated in the pathogenesis of allergy. When allergen proteins cross-link allergen-specific IgE on the surface of mast cells and basophils, allergic responses are triggered, and those can range from mild to life-threatening. The diversity and complexity of allergen epitopes, combined with the high affinity of secretory IgE-allergen interaction, have historically impaired the development of allergen-specific inhibitors of allergic responses. However, various herbal compounds have demonstrated the ability to inhibit immunoglobulin E-mediated allergic responses through different mechanisms. I'm not going to go through all of this. You guys can come and dig into it if you want to know more, but some of the molecular mechanisms behind inhibiting IgE involve the role of that FSIR1 signaling pathway, this kinase inhibition. Herbs can modulate the cytokine production, and I'm just going to scroll down to some of the herbs that have been found effective as immunoglobulin inhibitors. One of them comes from Kampo medicine. Kampo medicine is Korean traditional herbal medicine. And this one, I'm just going to go ahead and click on it. It's been used in Japan as an anti-allergy herbal medication for treating bronchial asthma. It includes 10 herbs, including Baikal skullcap. Now here at the top, you can click on this link to actually purchase it if you wish. And here's some studies about it. It suppresses type 1 and type 4 allergic reactions. Let's go back to another one is ashwagandha. Ashwagandha downregulates antigen-specific immunoglobulin E antibody responses in mice. Does it do the same in people? When they look at how it works, there's an assumption that it could transfer over to people. Then we've got this medication, this is another Korean herbal medicine that's demonstrated significant therapeutic effects for various conditions, especially atopic dermatitis and inflammatory diseases. And it's a fermentation of certain herbs with Lactobacillus acidophilus. The fermentation process improves the absorption and bioavailability of the active ingredients. Now, this is not available for sale. However, I've researched what's in it, and we also have instructions for fermenting herbs. So possibly we could make this ourselves. We just don't know the ratios. And here we discover the therapeutic actions of this formula. As far as fermenting it, you see here instructions on fermenting herbs, both fresh herbs and dried herbs. So possibly, understanding safety profiles, we could make something ourselves that is helpful. Okay, what else do we have? This Britannin is found in Inula japonica. Let's pull that up. That is called Oriental Yellowhead. Do you remember what else is in the Inula genus? Elecampane. And we know elecampane is wonderful for respiratory disorders. So possibly, see, does elecampane contain Britannin? Scrolling through this to see. I don't see it here, but it may be there. I'll have to do some more research on this and see if it also contains Britannin, because it's the Britannin that seems to be effective. And then we also see that chaga, strawberry extract, even apples, and then some of these various phytochemicals have been shown to help inhibit. So you could click on these and see where you can find them, like selenodin. Where do you find that? That's in Ashtaba, which we see supports the immune system, digestive health, and that is in the Angelica. So you can just explore around in here, and if you're interested in discovering how herbs can help with inhibiting immunoglobulin E, then there's plenty here to dig into. So we're working our way towards food allergies and food sensitivities, how they occur, and what kind of remedial steps can we take to either tone them down or eliminate them altogether. In our next class, we're going to take a deep dive into salmonella food poisoning. So stay tuned for that video. And in the upcoming class this Thursday, we are going to be starting our work with food allergies. I'll see you at the round table.